Hello, dear students of Solomec University. I'm glad to greet you in the first semester, in your new semester, first semester passed, and we start our online lectures of histology. So today I'll open three PPTs. First is study of tissue and introduction topic. Then we'll discuss epithelial tissue, and finally, in the end of this lecture, I'll explain you what is a gland. Okay. So histology. We discussed what histology is: the study of cells and extracellular components, and we can see it in light microscope way. And the collection of the cells and extracellular matrix is known as tissue. Histology definitely is the study of tissues. Despite their disparate structure and physiologic properties, all organs are made up of only four basic tissue types. And let's study these four tissue types. They are epithelium, connective tissue, muscle tissue, and nerve tissue. So epithelial tissue covers body surfaces, lines body cavities, and forms glands. In easy words, epithelium present everywhere encounter two different milieus. So inner milieu with outer milieu, blood with inner milieu, so outer environment our innate structures. So everywhere is epithelium. If you can reach it with your finger, somehow it is covered with epithelium. Okay, in more details, we'll discuss it later. Then connective tissue. It underlies or supports the other three basic tissues, both structural and functionally. So let's talk about connective tissue later. Muscle tissue. It is made up of contractile cells and is responsible for movement. So I hope you know what is a muscle. Yeah, and the muscles are contractile tissue which is able to slide their actin and myosin fibers inside their cells to perform the action. And we have different muscle tissue types, three muscle tissue types. Okay, we'll discuss it in proper slide. And nerve tissue. Nerve tissue is correlated with nervous system, and everything in nervous system, so every component of nervous system is composed of nerve tissue. It's our brain, spinal cord, nerves, and ganglions, so they are nervous tissue. Okay, let's talk about epithelial tissue first. Epithelial tissue is, as you can see here, so what is typical for epithelial tissue? This one is epithelium, this one is epithelium, and this one is epithelium. Seems like nothing is similar between them, but let me say it. So first of all, in all epithelial tissues, we have basement membrane. Basement membrane is a thin fibrous structure, so which is on which all epithelial cells lined up. So this is lamina. Actually, this is a straight lamina, so on which cells are resting. And so, for example, here it's visible much better. This line, so it's not properly visible. Yeah, it's visible only in the respiratory tract actually, but you can suggest that here we have some layer of some teeny tiny thin structure on which all the cells are sitting. So here, and here. This is a basement membrane. So basement membrane where all cells are sitting. Okay, that's first feature. Possession of basement membrane. Second, cells are very closely arranged to each other. One cell is followed by other cell. So one cell, and here very adjusted, very fine adjusted epithelial, another cell is present. Same as here. So you see one cell, and nearby you see borders of other cell. Then you see everywhere we have cell and cell connections, and here also one cell is neighbored by other cell. So this is typical feature of epithelial cell. Okay, third one is absence of extracellular matrix. So no extracellular matrix is present. So you see, so for example, compare this cell, this is typical fibroblast, and around we have extracellular matrix, which is not cell. So it's something which is synthesized by itself, but outside the cell. But what about the epithelial tissue? So actually, sorry, epithelial tissue possesses no extracellular matrix. So this thin part of the extracellular matrix is actually little fluid, which possibly present here. So also here you see, so extracellular matrix is almost invisible. Here it's totally invisible. Okay, so next, so here you can find the explanation of what we discussed. So let's move forward. It's a muscle tissue. So muscle tissue is everything what is contractile. As I said, it possesses actin and myosin fibers. So for example, this one is a skeletal muscle, also known as striated muscle. This one is a smooth muscle. Smooth muscle. You see this spindle-shaped cells without proper arrangement, without lines, so perfect lines as we see here, and spindle-shaped cells which are arranged in different directions. And here, it's a cardiac muscle. Cardiac muscle, it's they are so somewhere in between this and this. They are cross-striated like this, but a lot of features of smooth muscles, and we will discuss it in topic of muscle tissue. But keep in mind that there are three types of muscle tissue, and all of their function is contraction. So, skeletal, cardiac, and smooth muscle. Skeletal muscle equals to striated muscle. Smooth muscle is smooth muscle, yeah. And nervous tissue. Final nervous tissue, it has specific features, so absolutely unusual for other tissues. And you see that here we have axon, so axon, and around we have myelin sheath, and so on. So actually, nervous tissue is specific, and we will discuss it in proper topic. So because it's too high variety of nervous tissue appearance in different tissue types in different locations. So that's why here is shown nerve with a lot of nerve fibers. Nerve fibers are present in one nerve. So later we are going to describe it. Okay, of course you know the what is neuron. It possesses axon, cell body, and dendrites. So structure of this are discussed in I don't know in numerous times in biology. But still, in histology of nervous tissue, we will touch this topic and explain it in details. And finally, connective tissue. So, to ease your understanding of connective tissue, and I want to share with you that connective tissue is everything else, which is not typical for nervous tissue or muscular tissue or epithelial tissue. It is connective tissue. Connective tissue presence everywhere we have everywhere besides this tissue, so other tissue, because it's supportive tissue. It connects one tissue to another, and you cannot find the epithelium which is attached to muscle, or I don't know, muscle which is attached to nerve without any kind of connective tissue around. So connective tissue connects them together. And we have and connective tissue is very highly variable tissue. For example, blood is connective tissue. Blood, also bone is also connective tissue. Bone. So, and you see what is the relation between bone and blood. Actually, this is fluid. This is one of the strongest tissue in our body. But anyway, by classification, it is similar tissue. So that's why for now, just keep in mind that connective tissue is everything else. Okay, let's continue. Uh, Let me show you my face. Yeah. Okay. We start on nervous tissue, connective tissue. So, and typical feature of connective tissue is the possession of extracellular matrix. So that's the point. So what is different with other tissues? Usually, other tissues possess very little extracellular matrix, but connective tissue, almost in 95% consists of extracellular matrix. So we have few exceptions, but always extracellular matrix is a typical feature of connective tissue. And here example, yeah, so here you see the nucleus of cell. Cytoplasm of cell is also very shrinked, but everything, all these wires, all this tissue, all these components are extracellular matrix. That's the point of connective tissue. So that's why you can remember it in both ways. So connective tissue is everything what is not epithelium, nervous tissue, and muscular tissue. And also typical feature of connective tissue is possession of these wires. And also, what else I can say here? So in connective tissue goes blood vessel, blood vessel. Blood vessels are going in along the connective tissue, and everywhere we have connective tissue, it is tissue which is supplied by blood, and through the connective tissue, through this extracellular matrix, feeding is supplied to epithelial tissue, muscular tissue, and nervous tissue. All the nourishment, all the supplement goes through the connective tissue. Okay, let's move forward. So this slide, if you want to see the scientific explanation, scientific definition of connective tissue, so it's here. And in previous semester we stopped on this diagram. Yeah, so we stopped on neurulation process and embryo folding. So a little bit discussed about it, and further development is known as organic genesis. So if here we discussed the formation of three germ layers, later will be organogenesis, and and don't worry, embryology is not over. Sorry, I've been interrupted again. Okay, so and tissues which are formed here, so they are made by same layers of embryo. So as we know before, this is epidermis, ectoderm, mesoderm, and endoderm. So epithelium is predominantly made by ectoderm and endoderm. So, and almost all tissues which are made by ectoderm and endoderm, they're epithelial. Besides, of course, the neural tube, the neuroectoderm. So everything which is made by neural ectoderm, it is nervous system. But there are a lot of epithelial tissues which are made by mesoderm, but predominantly mesoderm is responsible for connective tissue and muscular tissue formation. So epithelium is a very little amount made by mesoderm. Okay, and here so I my request to you so to observe it again. So we a little bit discussed in previous semester, but so repeat is never too much. Okay, next topic is epithelial tissue. So and now we'll delve deeper in understanding of epithelial tissue. As I told you before, so epithelium is an avascular tissue composed of cells that cover the exterior body surfaces and line internal closed cavities. The internal closed cavities, so exterior body surfaces is skin, eye, yeah, and mucus, actually, the mucus of oral cavity and entire digestive tract, mucus of respiratory tract, mucus of urinary system, mucus of genital system also. So everywhere everywhere we have connection, direct or indirect connection to the external surfaces. It's covered with epithelium and internal closed cavities. Which internal closed cavities you know? It's a vascular system, it's peritoneum, pericardium, and also pleura. So pleural cavity. So peritoneal cavity, pleural cavity, and pericardial cavity are also covered with epithelial cells. And that's example of epithelium, which is made of mesenchymal cells of mesoderm. Okay, so body tubes, they communicate with exterior. So as I said, alimentary tract, digestive tract, yeah. Respiratory tract is our lungs and trachea, bronchi, and all of this. And genital urinary tract. So as I said, genital tract, for example, ovarian tube and even testicles, they are made up of epithelial tissues and lined up of epithelial tissue. Okay, epithelium also forms the secretory portion, parenchyma of glands. So all glands you know in the body, so they are made of epithelial cells. It's because of their specific feature to produce, so they have very active Golgi apparatus, which is able to produce secretory vesicles to remove it outside. In previous semester we discussed it, yes, that exocytosis is one of features of cells, and epithelial cells handle this feature in its best. And also we have specialized epithelial cells. Their function is receptors for special senses. It's smell receptors, taste, hearing, and vision. That's also feature. Okay. Functions of epithelial tissue are shown here. Protection, as well as present on the surface. We need to protect underlying tissues. Absorption. So as well, we connect the outer environment with the inner environment. Sometimes in some places we need to absorb tissue. So especially it's about respiratory, digestive tract. Yeah, in digestive tract we must to absorb tissue. Then secretion. Secretion is necessary to smear the surface of mucosa or even skin with mucus or sweat, respectively, and to make it smooth and easily passed by along these tubes. And in skin, so sweat serves to other functions, and we are going to discuss it in next year, I guess. But anyway, so you know that sweat is necessary for thermoregulation and of course exchange. So what's the difference between absorption and exchange? So actually exchange happens through the double membrane of the cell, so without interruption inside the cell. So it happens so to okay, example is alveoli of the lungs, and they possess exchange of oxygen and carbon dioxide. So this carbon dioxide and oxygen exchanged directly here in this alveoli of lungs. So because epithelial cells sometimes make in the very thin layer of cells, so they are possible for exchange in its best. Okay, about functions we'll discuss in direct explanation of types of epithelial tissue. Then characteristic features of epithelial tissue. I already named them, but let's make it in a more scientific way. Very cellular with a little intercellular space. So as I shown you before, so cell is followed with another cell, is neighbored by another cell, so very closely without interruption between them, without some specific extracellular matrix between them. Okay, and if you see so weighted cell or tissue, so you can be sure it's a epithelial tissue, usually avascular. So inside the epithelial tissue, you can never find the blood vessel. You can never find. So all the supplement of epithelial tissue with oxygen and nutrition goes through the connective tissue underlying this, so which is present under the basement membrane. And yeah, and of course, next point is that cells rest on a basement membrane. So here, as this is respiratory tract, it's I guess it's bronchi. So basis fragment is invisible, but I guess it's bronchi or trachea. So you see, cells are resting on very pretty thick basement membrane. All this pinkish colored structure is a basement membrane. In respiratory tract, it's pretty thick, and it's happened due to very high epithelium here. So that's why they need a very hard connection to the underlying connective tissue. And basement membrane is a source of attachment, so like anchor of the cells. And from another hand, here we have blood vessels. So it's not visible here, but blood vessels are embedding this connective tissue, and this blood carries the oxygen and nutritions, and through this connective tissue it passes towards the basement membrane, and through the basement membrane it passes towards the cell, and cell can live their life just because of the supplement through the basement membrane. Okay, cells show polarity, and this is part of a new chapter inside this chapter. So polarity is not about positive and negative charge, so not about it. Please don't confuse it. Don't confuse yourself. So polarity means that one side of cell is different from other side of cell. It is typical for epithelial tissue because in other cells usually they show in the so one side of cell, for example, this connective tissue cell, one side of cell is similar to another side of cell, so they have no differences, so they are almost similar. And even if we have differences, it's not related to the location of the cell, but here it's different. Okay, cells may display surface modifications. So may display surface modifications. For example, here you see surface modification is possession of cilia. It might be microvilli. It might be stereocilia. So in any way, so surface of the cells might be changed according to their function. If we need, for example, like here in respiratory tract, we need to read out the mucus, excessive mucus which is produced, for example, with infected with bacteria. So we need to possess cilia, which actively motile and remove this mucus outside. In case of digestive tract, we need microvilli to facilitate absorption. Yeah, and that's a point. Okay, we'll discuss it. Okay, sorry, I was interrupted. So let's continue. So cell polarity. So what is contributed as cell polarity? So I said you that I'll explain it. So cell polarity is made by differentiation of basal domain, lateral domain, and apical domain. These three compartments are making this epithelial cell as epithelial cell. So and let's start with the basal domain. Okay, no, let's start with apical domain. So apical domain is necessary to interaction with external environments. So any kind of environments which they are present. So they are necessary to so in case of this diagram, so they are necessary for absorption. Increase and here you see the apical domain modification, which is known as microvilli. So this is typical appearance of apical domain. So this microvilli is necessary to increase surface area, to make more plasma membrane, so to facilitate more intake of any goods and food to the cell inside. Okay, and then let's talk about lateral domain. About apical domain, apical domain compartments, apical domain modifications, so we'll discuss a little later. Then let's talk about lateral domain. Lateral domain is composed of two types of junctions. There are gap junctions and tight junction. This tight junction. So okay, tight junction. So let's start with tight junction. So predominantly. Obvious example of tight junction is present here. They are desmosomes. Desmosomes are made up of. Okay, I'll increase. So desmosomes, which are visible here, they are made up of two hemidesmosomes. So this part is produced by cell. This cell. So here we have nucleus, cytoplasm, but here it's skipped. But nucleus, with help of rough endoplasmic reticulum and Golgi apparatus, producing the hemidesmosomes and distributed towards the lateral domain of the cell. And these tight junctions, hemidesmosomes, attach to hemidesmosome of other cell. So here is another cell which produces their half of desmosome, hemidesmosome, and they stick together with help of cadherins, and that makes it like a knob-like connection. Knob-like connection. So let's return my face. Now, these knob-like connections, which stick the cells to each other in on the all length of the lateral connection between one cell and another. So that's just physical attachment. Physically, we tightly join the cells together. Okay, and group and actually also we have this dijunction more organized near the apical domain. So actually it's a border of apical domain and lateral domain. The structures which are known as which are two zonules, with the zonula occludens and zonula adherens. The two zonules are usually intermixed, but main idea of them that zonal occludens occlude the insulate the lateral domain from influence of outer environment. So here outer environment might be aggressive, so quite aggressive, and might create some injurious components, injurious compounds, like for example in stomach. In stomach we have very acidic and dangerous milieu, aggressive milieu. And to prevent leakaging of these components in a lateral domain, which might destroy the cell, so we have this special zonula known as zonula occludens. These junctions are not so tight, they are not so physically, they don't provide physical force for this connection, but it's like glue to stick it, to stick one cell to another cell, and it's like insulator. So here I can compare this zonula as so who boys understand. So when you work as a plumber, yeah, so you know that you have to smear the specific glue or silicone or any other glue stick like component to adjust tube to tube. Same is here. So epithelial cells stick to each other with help of the zonal occludens to prevent any leakages between the cells. And other part is the zonal adherence. Zonal adherence is actually made up of almost similar in structure as desmosomes. But they are going in a row. So a row of desmosomes from each side of the cell, which binds it together. It is not insulative, so water molecules can easily pass through it, but this zonal adherence, zonal adherence just mechanically, forcefully supports the zonal occludens. That's the main option of the main point of the zonal adherence. So to keep it together. So okay, let's return back to example of plumber. Yeah, when you stick it together with some glue, so this glue is not going to keep the insulation of this between the cells. But a possession of zonal adherence makes them glued together much harder, and they don't allow to unglue. So don't allow ungluing of the cells from each other. That's my option. Okay, that's apical domain, lateral domain, and tight junction of lateral domain. But also here inside the lateral domain we have gap junctions. That's what is shown here as green knobs. Gap junctions are junctions between cells with a hole inside it. So these holes are allows of passaging of water, saline solutions, and also some small molecular compounds like glucose, like I don't know, so any kind of carbohydrates, amino acids, nucleotides. So even sometimes fat molecules, but only little molecular weight fat molecules. They allow them transmission from one cell to another. So it is necessary to epithelial cells to possess them forward. Actually, when one cell might get a huge amount of carbohydrate, another cell might get more saline, for example. So salts absorbed here much better, and they need to exchange these components to make usual normal nourishment of both. That's the point of gap junctions. So they are present in different ways in different tissues, but main idea of gap junctions is communication between two different cells. Okay, and now finally let's switch to basal domain. Basal domain is it's a cell membrane with same hemidesmosomes as in lateral domain, but these hemidesmosomes are attached not to another hemidesmosome, but attached to the basal lamina. Basal lamina is the outer part of the basement membrane. So same stickers, same fibers which are present in hemidesmosome here, which join to another desmosome, are sticking here. This collagen type 4, this specific collagen type which adjusts cell to the basement membrane. So that's a difference. You see, apical domain has apical domain modifications. We'll discuss it later. Lateral domain possess tight junctions, so straight tight junctions, so which is named as tight junctions, and also desmosomes, which is also part of tight junction. So here it's composed of zonal occludens, which is work as glue stick, and zonal adherents, which work as the staples. Staples joined together. Gap junctions necessary for communication, and hemidesmosomes on the basal compartment, basal domain, joins the basal lamina. That's with the help of focal adhesions, so they are sticked very tightly. Okay, let's so specialization basal surface. Please, yeah, one thing. A basal plasma basement membrane is composed of two components, as shown here. It's basal lamina, but under the basal lamina we have reticular lamina. So for now it gives you nothing, but later in the connective tissue topic I will discuss it in more details. That the basal lamina is a typical feature of epithelial cells, relation of epithelial cells to connective tissue, attachment of epithelial cells to connective tissue. Okay. Lateral domain specialization. So here you can find the information about it, but let's move forward. This is apical domain modifications, and as I shown before, it's a microvilli. Microvilli is immovable, so they stand in one position without motion, and motion is possible only due to changes in terminal web. Terminal web is a component of cytoskeleton. So you remember actin with intermediate filaments, with microtubules which present in terminal web. They control the motion of microvilli. And inside this microvilli, so which sustain the position, sustain the shape of this microvilli, is composed of actin filaments. I hope you didn't forget this actin filaments from the cytology. Okay, this projections of cytoplasm, which is made by actin filaments. Okay, stereocilia. Stereocilia is, we can correspond it like same as microvilli, but extremely long microvilli. So, for example, this one, this average in microvilli average length is 1 to 3 micrometers, and in stereocilia it's up to 120 micrometers. So imagine the difference. And this stereocilia is present in inner ear and also present in epididymis. So in topics regarding this regarding male reproductive tract and inner ear structure, I will show the location of this stereocilia. But function there is not well known, also because it's immotile, so it's not present. It's not present for movement or something. Actually, we consider that the stereocilia is necessary for absorption, also to increase surface for absorption, but with specific features. For example, in inner ear, specific feature of the stereocilia is a sensation of vibrations which get into the inner ear, and actually you reshape the signal from the sound from the wave of sound into the neural sensation of the sound into your brain. Okay. uh, so in anatomy and in histology of ear, we'll discuss it in much more details. And then cilia. Cilia is a cilia is unlike stereocilia and microvilli, it is motile organelle. So it is composed of microtubules inside with also with actin molecules. So we have core of microtubules arranged 9 plus 2 pattern. This 9 plus 2 pattern is typical for motile organelles, and in epithelia it is example of this is cilia only. Later in pathology, in histology of organs, in system histology, we'll discuss it also in details. But now keep in mind that cilia can move to one side or another side, and with help of them we can facilitate the transduction of some material from one side to another. And examples of cilia in our body is respiratory tract, so where produced excessive mucus, and especially in time of inflammation, and we can remove this mucus with help of the cilia. Okay, and also it's present in female reproductive tract in fallopian tubes. And these fallopian tubes, which connect ovaries with uterine. So you know the function. Cilia moves the egg towards the uterus, and we discussed it in previous semester in fertilization and early development stages. So cilia helps to move the egg towards the uterus. Okay, and then let's move to the chapter of classification of epithelium. Epithelium is classified into this by these two features. Is by number of layers. We can see it might be unilayered, also known as simple, and multilayered or stratified. So here you see some examples. So this one is unilayered, one layer of cells present. And here is the example of multi-layer. You see 1, 2, 3, 4, few layers which are present here. So how they arranged is also a point of our further discussion. And also we can classify this epithelia by shape of cells. And cells might be squamous. So like here you see they're like flattened cells which are which covers the huge space. They're resting on basement membrane, and you see thickness is very low. And okay, I'll explain it later. And then cuboidal, so it's cube shaped. Columnar, like column, like parallelepiped shaped. It's like box shaped. And pseudostratified, so which is appeared as stratified, but actually all cells are resting on basement membrane. So you see unilayered cells, unilayered epithelia. Every cell, each cell of the epithelia is attached to basement membrane. But in stratified epithelia, so only first layer of cells is attached to basement membrane. Other cells are attached to cells which are attached to basement membrane. And about them, we'll discuss a little furtherly. Let's start with very beginning. So first is simple squamous epithelia. So as I noticed before, simple squamous epithelia are flattened cells which covers a very high territory, high space of a basement membrane, and they are present in areas where there no damage is possible to enter inside. So, for example, it is in our body cavities. So, in our so might be you revised in biology that we have body cavities like peritoneum, pleura, and also pericardium, where there are no frictions. And frictions are frictions are relieved by possession of fluids here. And that's why we don't need to prevent any kind of damaging just by fluid. Just fluid is saving us against the friction. Okay, then we move to the uh, another location of simple squamous epithelia is our blood vessels. Blood vessels are covered with a simple squamous epithelia. They prevent the leakaging of blood components outside. But sometimes in small vessels like capillaries, this simple squamous epithelia allows the passage of blood because of their little thickness, because of their thin appearance. Okay, then let's move to simple cuboidal epithelium. Simple cuboidal epithelium is cuboid in shape, and they are, and you can suggest, yeah, if this is squamous, appeared in not dangerous areas, so must be this cuboid. They're huge in size, much larger than here, and must be it's present in some areas where damaging is possible. So it is located in usually in ducts of glands. They're also located in some areas like so inside the glands also, and also present in kidneys, in convoluted tubules of kidney. So might be in physiology you discussed it. So convoluted tubules of kidneys, they have in the cube-shaped cells. Okay, let's move forward. So third example is a simple columnar epithelia. Simple columnar epithelia. You see, every cell is like a column, and cells are very closely attached to each other, same as here, same as everywhere. Nucleuses are located in one row. So in different cells it's different. Somewhere nucleuses are located closer to the apical domain, somewhere closer to basal domain. Usually it happens. It is located near the basal domain or closer to the middle of the cell. So functionally, you might see that this relation here is safest place, this less safe place, and this is located in less safe place. An example of this is the stomach, digestive tract, small intestine and large intestine, and also in uterine females. So this epithelia is very absorptive. Usually, it's present in areas where we interact with external environment, and we can easily trespass the molecule from this side to the deepest side. So because we have only one row of cells, yeah, the cells are they're high, but still it's possible to penetrate into the connective tissue, because all the exchange between these epithelial cells going through the basement membrane with connective tissue under it. You see everywhere, here and here and here, we have connective tissue with blood vessels under them. Okay, next example is pseudostratified epithelium. Pseudostratified epithelium is named so because of their appearance. So actually, if you are not well if you're not practitioner pathologist, you can confuse this pseudo-stratified appearance with a stratified appearance, with true stratified appearance. So that's why we named it as false stratified or pseudo-stratified. Pseudo means false. So this type of epithelium is present in areas with a higher risk of intervention. So with a higher risk of damaging and with less opportunities to absorb something. So it is located in respiratory tract, first of all, and male genital system. In vas deferens and in epididymis, we can find this type of epithelium. And one more thing about this epithelium is in this epithelium we have very abundant amount of mucus secreting cells. Mucus secreting cells are known as goblet cells, and we are going to discuss it in this lecture a little later in gland chapter. So, and this type of epithelia always, without exception, possess a very thick basement membrane, uh, as I shown you in real diagrams, yes, that in simple squamous epithelia, in cuboidal, and in columnar epithelia, you hardly can observe the basement membrane. Very hardly is possible, so because they have to attach only one row of cells. But here, so even this tall cell so has attachment here, and attachment space is very small, and we need to attach it very firmly. That's the point. Okay, usually surface of this epithelia is very mucous, and examples are once again respiratory tract, starting from trachea, actual nasal cavity too, nasal cavity, trachea, then bronchi and bronchioles. They are made up of this epithelium. Okay, and next example uh, is stratified epithelium. Stratified squamous epithelium. So it's known as squamous because cells here are also squamous on the upper position. So we can observe it as squamous type. So look on this half, please. The squamous cells, and it is almost similar as here, but by function they're absolutely different. So tremendously different. So first of all, let's figure out how the nourishment goes here. First of all, first row of cells is resting on basement membrane. These cells feed nourishment of the cell is very well. Oxygen supply is okay, so blood vessels are given enough food, enough oxygen to this first row of cells. And when cells are well supplied, they have a lot of nourishment. What they are going to do? Do you think? Yeah, they start to multiply themselves. This process is known as proliferation and cell division. So a lot of cell divisions, mitosis happens in this row, and this produce new cells produced here on this layer. They moving upside, and they form other layer, second layer. But this second layer has no proper attachment to the basement membrane. They have no attachment, and they have connection only via gap junctions to the first layer of cells. And of course, they get less nourishment, less oxygen, but still they get something. And they can also reproduce themselves, but with less rates, but they can multiply themselves. Okay, then next layer occurs. It is in this layer, so cells are suffering from hypoxia. They have not proper amount of oxygen. Proper amount of oxygen and no proper nourishment. And these cells are gradually dying. and they die gradually, and then upper side of the cells, upper side of this tissue is almost dead cells. And if we have any kind of how can you say, if we have any kind of injury here, might be not injury, just usual wiping. If you wipe something here, uh, something through it, so it will pass along with it. So must be this epithelia is present in areas where frictions are very common. And which areas with the friction? Of course, it's oral cavity. While we chew in some food, yeah, we eat something, so it might wipe something, yeah. And we need this epithelia to handle this, frictions. Okay, also it's present in esophagus during the swallowing, so we can swallow some root material, and also some cells from this from the upper surface might wipe away, and we need to reproduce them. And this epithelium is always forcing against the frictions. So if one layer of cells get fricted, so what get wiped, so what we need to do, we need to produce more and more cells instead of them. And this is a continuous cycle of changes in the stratified squamous epithelium. Okay, but here nearby, so attached to this, we have almost similar picture. Yeah, also we have cells on basement membrane, which get enough nourishment. Then second layer, which get nourishment from the basal cells, so cells on the basement membrane. Then cells upper, they suffer from insufficiency of food, and as upper we go, as less nourishment is available for them. And finally, dead cells, instead of shaving from the surface, instead of uh, extrusion, so they keeping together, as they became as a root, as a solid structure known as horn, horn-like structure. This uh, this is no, these cells are embedded with keratin. So keratin cells, keratinized cells, they are dead, but they're still cells. And that cell layer so prevents the it's present in areas when we need more protection than is available here, than is that we can offer here. So actually, keratinized surface is present in our skin, because we are taking, sorry for interruption. So here it's present on skin. So skin is an interaction with outer environment, the most outer environment, so which is most dangerous for us. And we need to save our body. So under this shield, so this shield with dead cell layer. So this dead cell also can shed out from the surface after due to friction, but it's hard to shed it out because they have some kinds of chemical susceptibility, so chemical fixation here, and no chemical can pass through this layer. So only with only very aggressive chemicals is possible to pass through this layer. This epithelia is absolutely non-absorptive. No absorption is possible here because we have too much layers of cells, and you can't absorb it. So unlike here, so for example here absorption is possible. Little absorption, yeah, not too much absorption. So only small molecules can pass through it, but they can. But here they can't. Okay, and then we move to the number 7. Number 7 is it's stratified cuboidal epithelium. Stratified cuboidal epithelium is located in also in ducts of glands, so for example in salivary glands, most outside located tubes are covered with this type of epithelia. It's necessary also to keep safe these ducts. It's much safer. This area of ducts is much safer than skin and mucosa of oral cavity, but still it's present. Yeah, also I forgot, sorry, a little referring back. This stratified simple squamous, stratified squamous non-keratinized epithelia without keratin layer outside is located in oral cavity, esophagus, anal canal, and vagina. Now, these four areas of our bodies, they are able to get fricted. Yeah, so in anal canal, it's during the defecation process. In vagina, during the sexual intercourse, and of course, we have to have the surface with possibility to be treated, to be healed by other cells. That's why we don't, without revenge, we just shed the cells out because we can reproduce them. Okay, and this locations we discussed here. Locations discussed. So location of this also discussed. It's ducts of glands of superficial arrangement. So in dangerous environments. So simple cuboidal is present in deeper layers of deeper levels of ducts, but this type certified cuboidal is located on the more dangerous situations. Okay, then stratified columnar epithelium. Stratified columnar epithelium is typical example of meibomian glands. Meibomian glands are glands which are located in our eyelid, so which producing the lipid-like, so fat-like secretion, which covers the eye during the process of sorry, I forgot that word. So by this process. Okay, I'll read it and I'll remind you this. Okay, and number 9 is it's transitional epithelium. So as here shown as 5, transitional epithelium. So it is located in urinary tract. Transitional epithelia, so in their features, is very familiar to the certified squamous epithelia, but difference in arrangement of desmosomes here. So the desmosomes here are much better arranged and much better connection between basal compartment and apical compartment of cells via the gap junctions, and nourishment of cells is sustained on a good level. This type of epithelia possess very insulative surface, and no absorption is possible, no absorption at all. So this type of epithelia is present in urinary tract, starting from kidney pelvis, overing with urethra. So in urethra. So one feature here is non-absorptive. So very important. So each of the epithelia is little absorptive. Okay, let's continue. Okay, and I stopped on the traditional epithelium. So it's present in urinary tract, and it's extensible, and it's and once again, sorry for repeating this again, it's impermeable for nothing. Actually, it's not absorbable, and it's pretty understandable. Yeah, that urinary tract is actually located in a very tight contact to urine. So what is already waste of our body, and we shouldn't and we mustn't absorb it back, and that's why this epithelium must be inabsorbable. And one little example, so what to confirm that transitional epithelium is not absorbable, it is that for example, so disorder known as cystitis. So when urinary bladder is inflamed. So in this case, it never ever became. It never gives in temperature to the increasing temperature to the patient. It never causes a fever. So because that's why cystitis, inflammation of urinary bladder, never cause the inflammation, never causing the fever. Okay, so if I'm not mistaken, that's it. So here we have explanations. So you can see it in PPT. I'll show to you later. Then let's move to next lecture. This lecture is about glands. So actually glands are so as I noticed before. So glands are epithelial tissue. So composed of epithelial tissue, and we have okay, and we have different types of glands, and by function they are divided into two types. There are exocrine glands and endocrine glands. So exocrine glands are we used to think about like exocrine glands are glands which produce secretion outside the cell, but actually every gland secretes something outside the cell. Okay, then others can say that exocrine gland produce secretion outside the body, but actually it's also wrong. Right way to say about it that exocrine gland is a gland which produces secretion on the surface of their cells, from the apical domain. You remember that apical domain, yes, that we discussed in epithelial cells. And when secretion goes outside or towards the apical domain to another milieu, so this is known as exocrine gland. Unlike this, endocrine glands produce their secretion through the basal domain towards the basement membrane and into the connective tissue below. Exocrine glands produce very abundant secretion. Usually it possesses a lot of water or any kind of lipid. So in case of sebaceous glands, and it's present in digestive tract, in respiratory tract, in urinary tract, and of course skin. Skin is also an example of exocrine gland. But in endocrine glands are producing hormones. So endocrine glands producing regulatory molecules which are produced inside the inside connective tissue, and later from the connective tissue it is transported to the rest of tissues by the blood. So that's the difference between exocrine and endocrine glands. But there are some transient types of secretions we have, so less abundant, less important for histological review, but very important in a way of physiology. So it's paracrine signals. Paracrine signaling, so it's when one cell is located somewhere in organ and produce their secretion outside the cell, so toward the cells which are nearby. So and they regulate the activity of cells which are nearby. They also produce it into connective tissue, but from the connective tissue it's not distributed towards the blood vessel, and side of action is directly there where paracrine signaling happens, when paracrine glands are producing their secretion. As example, I can say the inflammatory cells. For example, mast cells must be discussed in physiology, or you're going to discuss it next semester. During this semester, I guess. And this paracrine signaling hormones, they're acting on cells nearby. They are acting on blood vessels, like histamine, bradykinin, and other pro-inflammatory kinins. They're acting on cells nearby. They don't act on entire body. They act only there. And also we have autocrine secretion. It actually doesn't matter for us in histology, but it's very important in pathophysiology and in biochemistry, because some cells produce their secretion directly on the nearby to activize itself. So this strange mechanism, but it have less to do. They're present and we can't ignore it. So exocrine, endocrine mechanism, autocrine, and holocrine. I mean paracrine. Exocrine, endocrine, paracrine, and autocrine mechanism of signal. But in this lecture, we'll stuck on exocrine glands only. So exocrine glands are also classified itself into these two types. They might be unicellular and multicellular glands. So unicellular gland is a gland which possesses only one cell which secretes the secretion outside, so on the surface of itself. But multicellular glands producing the secretion accompanied with other cells, with other secretory cells. So, unicellular gland example is one of the best example is a goblet cell. Goblet cells are present along the digestive tract and respiratory tract, with producing mucus to facilitate transmission of food, transmission of and also moisturizing of the surfaces, so to facilitate proper adhesion and removing. Okay, uh, and multicellular glands are located in all other cases. So we will continue with the multicellular gland explanation. So multicellular glands usually form something like tube-like or other shapes, like invaginations inside the tissues, which are producing some secretion through the tube-like ducts. So that's the definition of multicellular exocrine glands. They produce secretion into the ducts. So first example is simple tubular, and you see shape is like this. So tube shape, and a secretory portion is here, and produces through the one duct. So we have one secretory portion and one duct. This is simple tubular gland. By the shape, you can say that cells here are cuboidal to columnar in shape, and they produce their secretion here. And usually secretion is waterose, so I mean aqueous, and very liquid, or might be mucus secretion. So it means it doesn't produce proteins. Usually, this type of gland produce only aqueous secretion, might be with polysaccharides. With polysaccharides, and actually these unicellular glands, like goblet cells, they are arranged in invagination here, and they produce this mucus, which is going to be extruded outside. Okay, and another example, I jump to simple acinar gland. Simple acinar gland is same as here, so they possess very small duct, one duct, and but secretory portion here is round shaped, so round to oval shaped. So I compare this and this. So, and cells here are acinar cells. These cells producing more proteinous secretion. It's known as serous secretion predominantly. Acinar glands are responsible for serous secretion, first of all. But it's examples of simple glands. Sorry. Okay, but we have variations of these glands. So actually everything else is just variations of these two types of glands. And let's start with tubular glands. So simple tubular glands, they are present in large intestine glands of the colon. They are also present in digestive tract, so somewhere in digestive tract, and also in uterus. They're located in uterus to produce some secretion there. Okay, another example is simple coil tubular. So you see, it's same as simple tubular, but it gets coiled. So this curvature happens, and actually it goes few times, a lot of times, and it's a specific type of tubular glands. They're located in sweat glands and merocrine sweat glands. We'll discuss what means merocrine, but this also sometimes it's named as ecrine, but more commonly it's used like merocrine sweat glands. just in this table. It's somehow they named it as eccrine. These coiled tubular glands are located in the skin, and they produce sweat on our surface. Okay, then another example is simple branch tubular glands. So you see what's the difference between simple tubular and simple branch tubular. Simple tubular possess one portion and one duct, one secretory portion, one duct. But here we have a bunch of secretory portions, yeah, and only one duct. So you see, it gets a little more complicated. So it's like branches. Yeah, it possesses branches, but finally it leaks into the one duct. Examples are stomach is mucus-secreting glands of the pylorus, and also it's present in uterus in endometrial glands. Sometimes we have simple tubular glands there, but predominantly we have the simple branch tubular glands in the endometrium. So endometrium is uterus, of course. Okay, uh, and another example is the compound tubular gland. Sorry, a compound tubular gland. So unlike branch tubular gland, so it possesses a system of excretory. Uh, so uh, sorry. Yeah, they possess a complex structure of tubes which release the secretion outside. So once again, check this out. Once again, here is branched. So just one branch, so one sorry, one duct and few secretory portions. But here we have a system of ducts which are going to another duct. And we have a large variety of these glands, but most common example is some mucosal glands of Brunner in the duodenum. And the more obvious example is our salivary glands. It's the sublingual salivary glands. They are predominantly this compound tubular in shape. Let's talk about acinar glands. Simple acinar glands are located in urethra, in paraurethral and periurethral glands. Their simple acinar glands are always produced by transitional epithelium, as we discussed before. So invagination of transitional epithelium forms the simple acinar glands shape. And so I already said it's paraurethral and periurethral glands. They are necessary to help the transmission of sperm along the penis. Okay, then we have branched acinar glands. Branched acinar glands are they are present same as here, same as simple branch tubular, but this is simple branched acinar. So we have one duct and bundle of acinar glands. So these glands, unlike simple tubular glands, they are also round in shape, and cells are acinar in appearance, and they usually produce protein-like secretion, proteinous secretion, and this is known as serous secretion. So you see, simple tubular glands usually produce mucus secretion, and branched acinar glands, so all acinar glands are producing serous secretion. Once again, mucus secretion is composed of water and polysaccharides. Acinar glands composed of water and protein. That's the main difference between these two types of glands. And doesn't matter it's simple or branched or complex. So location of branched acinar glands is the stomach. It's present in so this obvious mistake is serous secreting glands of cardia, which secrete the enzymes of the stomach. And in skin, we also can find branched acinar glands, which are located in sebaceous glands. Sebaceous glands. So sebaceous glands, which produces the sebum, this lipid-like secretion on the skin. Okay, and compound acinar glands. Examples of compound acinar glands are not so abundant. This example is pancreas. So you heard about the pancreas is a mixed gland. Yeah, so it produces so as endocrine and exocrine secretion. But now we're discussing the exocrine portion, and exocrine portion of pancreas is composed of compound acinar glands. And they produce, I remind you the function of pancreas, they produce enzymes, and enzymes are proteins. That's why it means it is serous secretion. Yeah? And compound acinar glands is also responsible for serous secretion. Another example of compound acinar gland is our parotid gland. Parotid gland, which is located near the ear, which produces the saliva. And saliva, which is saliva of parotid gland, is much thicker and possess enzymes, also enzymes which help us to digest food directly in our oral cavity. And we have a few examples of compound tubular acinar glands. Compound tubular acinar glands are glands which are composed of both acinar glands and tubular glands. And it's like a mixture of these two types of glands. And then these examples are so very one of the most common example is submandibular salivary gland because it produces both and enzymes and lysozyme and also producing the aqueous mucus secretion. Okay, and final, so I hope you're not sleeping now. So please pay attention to this point. As there will be a lot of questions about this topic. Is types of secretion. So how we can secrete the secretion outside the cell. First example is a merocrine secretion. Merocrine secretion handles in this way. So Golgi body, so you remember the function of Golgi body, it produces, so with help of rough endoplasmic reticulum and smooth endoplasmic reticulum, it produces a lot of cell membranes. And in these membranes is collected some secretion. It might be mucus or serous, doesn't matter. But inside it we have some components and it detaches from the Golgi complex, float towards the apical domain, fuse with the apical domain, and then finally release secretion outside when by usual process known as exocytosis. This we also discussed in previous semester of histology. If you forgot about this, please open my lectures in previous semester. Apocrine secretion when also Golgi complex takes part in it, but goes in opposite way. So Golgi complex, along with the rough and smooth endoplasmic reticulum predominantly, produce some droplets of lipids, and so produce droplets of lipids or some other components, and collect it into near the apical domain of the cell. And as well as is it collected, and when this here is collected enough components, so some part of cytoplasm just pinched off of the cell. It's pinched off of the cell and moved down. So examples of this apocrine secretion is lactating mammary gland. So lactating mammary gland means during after the pregnancy, so after the delivery actually. So we need to feed our babies, and apocrine secretion helps us with it. Okay. And final type of secretion is known as holocrine secretion. Holocrine secretion is when a cell commits suicide in the end. So everything starts similar to apocrine secretion. Also Golgi apparatus producing some vesicles inside the cell, so composed of also predominantly, usually it is fat-like secretion, fat-like components. They're collected inside the cell, along the entire cell, and then finally cell bursts and dies. Of course, cell dies after this. It sacrifices itself for secretion. That's the difference between holocrine, merocrine, and apocrine secretion. Example of this is sebaceous glands. Sebaceous glands are also, once again, it's glands present on skin which produce sebum, oil-like secretion, which makes our skin with a fat-like surface. Okay, and once again, I want to remind you we have two modes of secretion. It's serous secretion and mucus secretion. And then three types, three mechanisms of secretion. It's merocrine, apocrine, and holocrine. Differences are discussed below. Okay, thank you for attention. I hope, I don't hope, I sure that you learned it until this moment. If you learned until this moment, please type me in direct in my WhatsApp and say to me that you reached this part. Thank you. It will be extra points for to you. Thank you. Bye.